Evolution is based on four major ideas. None of them have ever been observed.
Before evolution can even start, you have to have that first cell. Nobody has ever made a cell in the laboratory.
And these types of discussions on internet forums with, internet atheists, they want to use evolution weakly defined. They're not even talking whether they're not clear whether they mean change, whether they mean common ancestry, whether they mean mutation and selection or some other proposed evolutionary mechanism. It's a very hand-wavy explanation.
The video you're about to watch features Professor Major Coleman, who lays out the four major pillars of evolutionary theory and the problems therein. He makes the audacious claim that none of these four basic claims have ever actually been observed. Rather, an entire generation or two or three have been indoctrinated to believe that evolution is evidence-based and is a sufficient explanation for life as we know it. Then I'm going to weave in a few critical portions of interviews that I've conducted over the past year with experts to sort of show the receipts, as they say, and hopefully make it crystal clear that the narrative that we've all been presented in school is largely mythological. Let's get into this.
Evolution is based on four major ideas. None of them have ever been observed. Number one, that life comes from non-life. We have not one single example of life coming from non-life. Number two, that all the life forms that we have today came from single-cell life forms. Many people do not know that while we do have single-cell life forms that we call bacteria, we have no two-celled life forms, we don't have three-cell life forms, we don't have four-cell life forms, we don't even have five-cell life forms. That is, we go from single-cell bacteria to complex life forms. So there's no evidence that all life comes from single-cell life forms. The third postulate of evolution is that time and chance drive evolution forward, of course, and time works against evolution. The chance of one simple 100-unit protein coming together by accident this is a very big number. Time and chance make evolution absolutely impossible. The fourth postulate that evolution is based on is that what we see today, minor genetic variation in species, is evidence of macroevolution. So because cats are different colors, dogs are different sizes, that means that fish turned into cows. And of course, we know that that's not true because Mendel proved that all the genes that exist today, you can shuffle them around, but there are no new genes that created.
Okay, so let's just go to that first claim that life comes from non-life. Is that evidence-based? Is that scientific? Or is it not? Here's Dr. James Tour of Rice University answering this question and laying the groundwork for where this current conversation sits. Let's get into it.
Before evolution can even start, you have to have that first cell, which is an entity, which is what's called a lipid bilayer. It's like a sac. And inside of that, although the sac is extremely complex, it's not just like a burlap bag or something, it's extremely complex. And then you have all the inner workings of that. Nobody has ever made a cell in the laboratory. Nobody has even taken all the components of a cell. So if you just take a living cell, you deconstruct it into its fundamental pieces and try to put that thing back together again. Nobody's ever done that. Nobody's ever even come close, and nobody even has an idea on how to do it. And every year, that target of making a cell becomes more complex, not because the cell has changed, but because we realize more and more the complexity that's in a cell. It used to be thought that that's just a blob of protoplasm, but it's not that at all. It's a whole hierarchical system of operation of molecules working together. So nobody knows how to even take the basic constituents and put that together. Well, how about making the basic constituents in a way that an early Earth would have had to make it? This gets back to the origin of life. How do you make those basic constituents? Nobody knows. And so the building blocks of the building blocks, people have some certain ideas, and it's rough. The chemistry is very, very hard. And they'll use modern techniques, and they'll try to insert themselves. They'll use what's called relay synthesis to try to ease the problem. All these things that an early Earth never would have been able to deal with. And then once they get these basic raw building blocks of say, a sugar, an amino acid, and a nucleotide, now you've got to hook those things together. You have to make strings of them. Nobody knows how to do that. That's in fact the basis behind my challenge. Nobody knows how to make those strings. And because you have to make those to get your basic building blocks of a cell. Nobody can even make those yet. And then there's a much bigger inherent problem, which is you need the informational code. It's like if you had a computer, but you had no software. Where is the software that's going to run these things? Nobody understands where the code is that encodes this thing. So these are all the problems with Origin of Life. And I'm just here pointing out the obvious. Truly, I'm pointing out the obvious. It's not like, wow, Jim Tour sees something that nobody else sees. No, I see what everybody else sees. Mm-hmm. Every other synthetic chemist, every other organic chemist sees every problem that I see. It's just that before I started studying in this area, I took it for granted what these guys said. You know, they would say all sorts of things, and you take things for granted because you think they understand. But then when you look in it, you see that this is a house of cards.
So there you have Dr. Tour in a pretty dramatic way, calling it a house of cards, saying that there's this really, really big problem, which is the origin of life itself, that is necessary to even begin to talk about evolution, because evolution requires there to be a cell before you can even talk about evolution. And so the idea that all life as we know it came from the so-called primordial soup is literally mythology, is literally a speculation that is not evidence-based at all. Now, let's go to Dr. Stephen Meyer answering the question of, can new biological forms come about through the evolutionary process?
The main thing evolutionary theory was designed to explain was the origin of new biological form. 2004 book with MIT Press lists a whole category, Stuart Newman and Gerd Muller, and they have a table in their introduction to the book, and they list all the unsolved problems of modern evolutionary theory. One of them is the origin of biological form, the very thing that Darwin was supposed to have explained. Let me just, I made both these arguments on the Rogan podcast, but let me make them again. Mm-hmm. Okay.
Chuck Norris doesn't do push-ups, he does earth downs. But what makes Chuck Norris so unstoppable? Well, the answer is obviously a lot of things, but there's one that I want to tell you about today. So here's the quick backstory. Chuck Norris reached out to me and he was like, do the people on YouTube know about? And I was like, what is? He could tell that I was not getting it, so he was starting to get frustrated. I started to get nervous, so I was like, all right, Chuck, I'll hear you out. What are you talking about? He started to talk about the reasons why some people age faster than others. And I was like, Chuck, please, I don't want to be on your bad side. Could you slow down a little bit? Can you just make your own video where you unpack all of this so that the people can hear from you themselves, so that I don't have to be the middleman? I don't want to triangulate all this, but I also don't want you to be mad at me. I don't want to cross you, Chuck. Can you just spell it out? So if you guys would hear Chuck Norris explain all of this for himself and also save me from the imminent danger that I seem to be in, go to chuckdefense.com/dose. That's chuckdefense.com/dose, or click on the link below. Now back to the video.
Mutation selection mechanism. Well, let's start here. If you want to build a new organism, we now know you have to have new information. It's just like in our computer world. If you want to give your computer a new function, you have to provide new code. So if you want to build a new animal form with new anatomical structures and therefore new types of cells, then you're going to need new proteins to service the cells, the new cell types. And so at the very least, you're going to need a lot of new genetic information to build those new proteins. So where does that information come from? Well, the standard Darwinian explanation is that new variation, biological variation, making new form possible, arises as the result of randomly arising mutations that are then, if successful, preserved and passed on by natural selection. But since the 1960s, we've become really since 58 in Crick's sequence hypothesis, but that took a while to confirm. Since the 60s, as it became clear that mutations were operating, if they were operating, they were operating on the sequence of characters, the nucleotide basis in the DNA. Crick's sequence hypothesis was that DNA contains digital code that is the nucleotide basis, the subunits along the DNA spine are functioning like alphabetic characters or digital characters in a section of machine code. They're providing instructions for building proteins. So since that realization has arisen, we realize then that what mutations would be doing would be changing those arrangements of ACs, Gs, and Ts, the digital characters in the quaternary genetic code. Now, we have analogous experience of those of that type of information carrying system, and that is in our computer world. We have digital software. Bill Gates' DNA is like a software program, but much more complex than any we've ever devised. Richard Dawkins has acknowledged that DNA is like a machine code. Leroy Hood says DNA contains digital code. So what happens when we randomly change a digital bit string in a section of functioning computer code? If we randomly change the zeros and ones, what happens? Well, two possibilities. Well, here's the question. Do we degrade the function of the code before we ever get to a new stretch of functional code? Or is it possible to gradually, by random means, change one section of software and develop a new, say, program or operating system? If I ask that question hypothetically to a group of software designers, they all laugh. It's obvious what happens, that those changes will degrade the existing function long before you'll come up with some fundamentally new function that a new stretch of code would be required to produce. Turns out the same thing is true in the biological realm, that the sections of the arrangements of A's, G's, and T's that will code for functional proteins are extremely rare among all the possible ways of arranging those characters. Exponentially rare. For one short protein, a colleague of mine, Douglas Axe, who worked on this problem at Cambridge for 14 years, determined that the ratio was 1 over 10 to the 77th power. For every string that will provide a stable, function-ready, function-capable structure called a protein fold, there are 10 to the 77th ways of arranging those same characters that will not produce such a thing, that will give you gibberish. So what that means is then, as you start to change the digital characters, the A's, G's, and T's, you are overwhelmingly more likely to fall into a functionless abyss than to maintain function. And as other scientists have done experiments, they're called site-directed mutagenesis experiments, they found that this is absolutely true. There's a scientist in Israel named Dan Tawfik, who died fairly recently, very tragically, but did some very important work on this same question. And he started randomly changing the characters in the DNA to see what type of effect it would have on stable protein folds. Could he generate a new protein fold from a pre-existing protein fold? And he found that within a very small number of mutations in his experiments between 3 and 15, the stability of the protein fold was lost. The thermodynamic stability was lost. It would no longer fold into a stable structure. Therefore, it was no longer possible to perform a function. So he said that the protein folds, which are the fundamental unit of biological innovation, they are isolated from one another in almost like galaxies separated from one another. So whereas we can get modifications of function and even some new function arising within the structure of a stable fold, getting from one fold to another is effectively biologically, it's extremely implausible. We have no Darwinian account of how that happens.
Hey, I wanted to let you guys know that I'm launching the weekly Dose of Wisdom newsletter. This is going to be totally free. It's going to contain a section called Food for Thought, which will contain an informational blurb tying in either something from science, philosophy, or theology. Then there's going to be a section that will showcase examples of how God is working in this community. I hear so many cool stories from you guys about how God is working, testimonies, and I wanted to put all of that in one place. Finally, there's going to be a mini devotional that'll be based on scripture, and also I'll provide some commentary. Also, you'll be happy to hear that there is now a free tier of the Wisdom Society called the Today Group, where you can receive prayer and join in fellowship with believers from around the world. And so if you're interested in that, the official launch meeting is March 5th at 10 a.m. Eastern Standard Time. Check the link in the description below to sign up for both of these free, totally free offerings today. Now back to the video. Thanks, guys.
Now, in response to this argument that I made on Joe Rogan, we had a young postdoc at University of Cambridge, whom I happen to know, challenge me by saying, well, no, in the COVID proteins, one of the proteins that's involved in the COVID virus, there's a protein that has accumulated 37 or 33, I can't remember, mutations. And he said, aha, Meyer said it was between 3 and 15, but that misunderstands the argument. because the argument is not about how many mutations can accumulate in a fold. It's about what will happen to the fold as mutations accumulate. And the claim is that as mutations accumulate, you will eventually drop into a non-functional abyss before you'll ever produce a new protein fold. And it happens in the proteins that Tawfik examined that the threshold was about 15. But we've never said or denied that you might accumulate more mutations, just that as you accumulate the mutations, you will inevitably lose the structural stability of the protein in question before you generate a new protein fold. And the counterexample that was offered by this young postdoc actually proved that point. The protein that was involved in COVID has 1,200 amino acids. The 30-some changes did not actually change the existing fold. It just accumulated random changes that had no effect on the fold structure. It hadn't yet degraded. But typically to get from one fold to another, you need about a 75% difference in sequence, and 35 or 37, whatever it was, divided by 1,200 is about 3%. So this was a 3% sequence change, not nearly enough to have a hope of changing a fold. And so, and the big point was, this accumulation of mutations did not change the fold structure. And that's the fundamental problem that we have gone to the mat to identify, is that the accumulation of mutations will degrade the stability of a fold long before they'll ever produce a fundamentally new fold. And as long as I'm on this track, there's an additional reason for that. It has to do with the physics of fold stabilization or destabilization. As you accumulate those mutations, the fold will destabilize and lose structure, and at that point, you can't get it back simply by randomly mutating.
And what is sort of like the philosophical significance of everything that you just unpacked there, just to pin that?
Well, it's the scientific significance first, and that is that neo-Darwinism, which relies on random mutations and natural selection, cannot give an account of the most fundamental unit of change in biological history. If you want to build a new animal, you have to have a new body plan. To build a new body plan is a unique arrangement of body parts and tissues, new anatomical structures and tissues. Well, if you need new tissues or anatomical structures, to build those, you need new types of cells. But new cell types require unique proteins. If you want to build a gut, for example, you've got to have digestive enzymes. But proteins are built from the information in DNA. And so to build anything in biology, at the very least, you have to have new instructions in DNA that are capable of building new types of protein folds, capable of new types of protein functions. And if you can't explain that, then you haven't really explained anything. And to extrapolate beyond the strictly biological and anatomical, to say, well, we can explain human behavior or the origin of morality, or the origin of criminality, or by in Darwinian means, is, as I say, it's laughably facile. And these types of discussions on internet forums with internet atheists, they want to use evolution weakly defined. They're not even talking whether they're not clear whether they mean change, whether they mean common ancestry, whether they mean mutation and selection, or some other proposed evolutionary mechanism. It's a very hand-wavy explanation.
Hey guys, if you like this type of content, I really genuinely think that you would like the Daily Dose of Wisdom podcast. We've had guests like John Lennox from Oxford, Stephen Meyer from Cambridge, and many other amazing guests. Check it out on Spotify and Apple. Also, I spent 2 years making a documentary film that is now totally free for you to watch. It answers the question, what are the biggest obstacles to belief today? Go to dailydoseofwisdom.com to check it out. You'll also see merch there, details about our online community where you can pray, share, and study with believers from around the world. It's called the Wisdom Society. Personally, I've been loving it. Check it all out at dailydoseofwisdom.com. Thanks for watching. See you guys in the next video.